we are seeing a shift in biology right now that is genuinely just breathtaking.
Oh, absolutely.
You have these technologies like StereoSec, Xenium, and now this new high-resolution Visium HD. They're just pushing the frontier of what we call spatial transcriptomics.
Right.
We're not just looking at snapshots anymore. We're building entire atlases, blueprints of tissue with millions of spots, millions of cells all mapped out.
It's the next revolution, for sure. But, that scale brings a huge burden.
How so?
Well, if you have an atlas with millions of data points, and the analysis you need to run takes days for just a tiny piece of that.
Yeah.
You've hit a wall.
Yeah.
A massive computational bottleneck.
And the key analysis here is something called cell type deconvolution.
That's the one.
Right.
Yeah.
So deconvolution is just this essential task where you have a tiny spot of tissue and it's got a mix of cells in it.
Maybe some T cells, some tumor cells.
Yeah, some fibroblasts.
Yeah.
And you need to figure out the exact proportion of each of those cells, right, using a single cell reference.
Exactly. And historically, the methods that give you the best accuracy, these really sophisticated Bayesian approaches like cell-to-location, they are just excruciatingly slow.
They're great, but just not built for this new scale.
No. They make a million-spot analysis basically impossible.
So for our deep dive today, we're going to unpack a new framework. It's called FlashDeconv.
It takes a pretty radical approach.
It uses a mathematical shortcut structure-preserving randomized sketching to get that same top-tier accuracy, but it just blows the doors off the speed. Orders of magnitude faster.
But here's the really critical part. This isn't just about speed. FlashDeconv solves a really deep, a systematic problem that plagues almost all the old methods.
Okay.
Especially when you're dealing with rare cells. The fatal flaw is that they systematically miss the signals, the ones from rare but biologically critical populations.
Okay, let's unpack that right now, that core conflict. Why does the standard way of doing things, analyzing these huge genomic data sets, why does it blind us to the most important rare players?
Well, it all comes down to how we've traditionally defined what's important in a data set.
Okay.
When bioinformaticians do dimension reduction, and you have to, with 20,000 genes, they almost always rely on one thing, variance.
Variance, which is, essentially how much a gene's expression changes or fluctuates across the whole data set.
Exactly.
We can think of it as measuring how loud a gene is.
That's a perfect analogy. And it feels intuitive, right? If a gene is changing a lot, it must be doing something important. But here's the problem. That loudness is just naturally dominated by the most abundant cells in your sample.
Ah, so if I have a massive structural part of the tissue, like, say, all the epithelial cells in the gut, or a big population of glia in the brain, their common genes, even if they're only changing a little, are just going to overwhelm the variance rankings by sheer numbers.
Exactly. Think of it like a concert hall. You have 80% of the audience singing along to a popular song. They're loud, right? Even if they're not singing anything unique. Meanwhile, you have the rare cell types. Maybe cancer stem cells making up less than 1% of a tumor. Or these really specialized immune cells, their unique molecular signal gets lost.
Because their population is just too small.
They're mathematically quiet. And so variance-based methods just systematically underweight them. They filter them out.
So it's that classic paradox. The thing that's rarest is often the most important, but it's the first thing we accidentally throw away. So this is where FlashDeconv makes a big pivot.
A huge one.
Instead of variance, it uses something called leverage scores. So what are these? How do they capture what's important without just measuring volume?
Leverage scores are all about measuring distinctiveness. Forget loudness, forget size. Imagine you're building a complex geometric structure, right?
Okay.
Variance measures the size of your biggest, most common building blocks. Leverage scores measure whether a gene defines a crucial structural direction in that high-dimensional space. It's looking for the unique fingerprint of a cell type, and it doesn't care how big the crowd is that's using that fingerprint.
So it's about structural definition, not statistical popularity. The source material had this brilliant piece of evidence for this. They called it the abundance invariance stress test.
Oh, that stress test is the mathematical proof of concept. It's beautiful. The researchers, they took mouse brain single-cell data and they just artificially destroyed the abundance signal.
What'd they do?
They took the dominant cell type oligodendrocytes, about 27% of the total, and they just systematically downsampled it.
Wow.
They reduced its frequency by over 60-fold. All the way down to just 0.4%.
So they intentionally made the loud population quiet, just to see what happened.
And the results were so stark, they tracked the ranking of the marker genes for those oligodendrocytes, the variance-based ranking. It just degraded linearly. It fell apart, deteriorated twofold as the population shrunk.
But the leverage scores?
The leverage score ranking remained stable the whole way through. It just didn't care that the population was now almost invisible.
That's the mathematical decoupling you want to see. It proves you can separate a gene's real biological identity from just, its parent cell type's frequency.
Right, and you can visualize this really beautifully. If you plot all the genes on what they call the variance leverage plane, the crucial quadrant they found contained what they call the Golgi genes.
Golgi genes.
Low variance, but high leverage. These are the exact genes the old methods filter out, but they're the ones that are structurally informative.
And what kind of genes are we talking about? What's in that Golgi set?
You find these classic low-abundance vascular markers like ChelidM5 and RGS5. These are genes that define really rare cell populations that are essential for structures like the blood-brain barrier.
And what about the opposite? The stuff the old methods prioritized?
Those were the noise genes. High variance, low leverage. 35% of those were just unannotated transcripts, statistical junk, really, compared to only 6% in the Golgi set.
So the old way of doing feature selection was literally prioritizing noise over crucial biological markers. Did this actually play out in the physical tissue?
It did, emphatically. The functional validation was a slam dunk. The Golgi genes were hugely enriched for vascular development pathways with incredible statistical confidence.
And on the actual images?
When they visualized them on mouse brain sections, these Golgi genes perfectly reconstructed clear, recognizable blood vessels, the anatomical structures. The noisy genes, on the other hand, just random speckles. Zero significant functional enrichment.
Okay, let's take a quick pause here and just recap. We've established that the big challenge in spatial biology isn't just the sheer volume of data. It's this systematic bias against rarity.
Exactly. Gene loudness or variance is just a poor substitute for biological importance.
And FlashDeconv gets around this by using leverage scores. It isolates those Golgi genes, the rare markers that define a cell's identity, even when they're super scarce. So we have the what and the why. Now, for the big question, the how. How did FlashDeconv take this mathematical insight and build a whole framework that not only preserves these signals, but can scale up to analyze millions of spots on a regular computer?
This required a complete re-engineering of the whole pipeline, and it's founded on 3 key computational choices that all prioritize linear scalability.
Okay, let's start at the beginning. Pre-processing. You said they had to make a pragmatic compromise on the data normalization.
That's right. So we're getting ready to compress the gene space, right? We're going from, say, 20,000 genes down to just 512 dimensions.
Wow.
So the researchers, they chose a log CPM transformation, even though other methods like Pearson residuals are often, statistically better for count data.
Why that trade-off? Why log CPM?
It was purely for speed and stability. Log CPM has what's called a bounded norm. In simple terms, it puts a ceiling on how massive any single gene's expression value can be.
And that matters because
it prevents these extremely high expression but very common housekeeping genes from mathematically just dominating the whole compression process. And that's essential for the core engine, this structure-preserving randomized sketching.
Okay, let's talk about that engine. If you're taking 20,000 genes and just brutally squishing them into 512 dimensions, this sketch, how on earth do you guarantee you don't lose the precious, rare Golgi genes?
This is the genius of the leverage-weighted sketching. So think of your 20,000 genes as letters you need to file into only 512 mailboxes.
Okay, so a lot of letters are going to end up in the same mailbox.
Hash collisions, they're inevitable.
And in a normal random system, if a common flyer, a high-abundance, low-leverage gene collides with a critical, rare letter, that common flyer signal just drowns out the important one.
Precisely. FlashDeconv uses a sparse count sketch matrix for the compression. But here's the key. The projection is weighted by those leverage scores. We give the rare, high-leverage genes an asymmetric gain. We basically print their letter in bold and put it in a thick envelope. So when a collision happens, that weighting ensures the important rare signal is preserved over the noise from the abundant genes.
So clever. Okay, so we've compressed the G dimension, the genes, from 20,000 down to 512. But the N dimension, the number of spots, is still in the millions. That's where the memory crisis happens, right?
That is the crisis, because we need to incorporate the idea that tissue is continuous. Cells aren't random, they organize smoothly. This is called spatial regularization.
Okay.
Older methods, when they try to do this, they often calculate these dense covariance matrices, and that leads to a quadratic memory cost, O of n squared.
O of n squared. For those of us who aren't mathematicians, why is that so crippling when n is in the millions?
Well, it means if you double the number of spots, the memory you need doesn't double, it quadruples. So if you have 1 million spots, you're talking about 1 trillion calculations just to figure out how every spot relates to every other spot.
That's just impossible. You'd need a supercomputer.
Right, and massive amounts of time. It's prohibitive.
So how does FlashDeconv get around this O of n squared bomb?
They use a sparse graph Laplacian regularizer.
Okay.
So instead of calculating the interactions between all million spots, they only calculate the connection to the k nearest neighbors, usually at 10 neighbors per spot.
It's like a social network. You don't map your connection to every person on Earth, just your 10 closest friends.
Exactly that. And because that graph is sparse only local connections, it scales linearly, O of n times k, both in time and in space.
And that linear scaling is the big advantage.
It's the defining advantage. I mean, look at the numbers. FlashDeconv can process 100,000 spots in under 4 seconds. They deconvolved a simulated 1 million spot dataset in about 3 minutes.
And the old methods?
Probabilistic ones like stereoscope, they need over 8 hours for just 10,000 spots.
So this is no longer some academic exercise that needs a university supercomputing cluster. FlashDeconv makes atlas-scale analysis millions of data points possible on your standard off-the-shelf computer with 32 gigs of RAM.
And crucially, the speed comes with no loss in quality. It matches or even exceeds the top-tier methods for detecting rare cells.
All right, let's do another quick recap. To summarize the architecture, FlashDeconv gets its speed from 2 main things. First, compressing the gene space using that clever leverage-weighted sketching to save the rare signals.
Right, from G down to 512.
And second, handling the massive number of spots, n, using a sparse graph. That avoids the memory-killing O of n squared costs of the old methods.
And that combination fundamentally changes what you can even look for.
Which brings us to the next question. Beyond the speed, what real biological discoveries were made possible by its ability to resolve these rare cell signals at high resolution?
Okay, let's start with a clinical application. Human ovarian cancer. The researchers looked at 6 patients, and they were stratified by how well they responded to treatment.
And the rapid turnaround let them do what? Validate biomarkers almost instantly.
Instantly. The entire cohort, all 6 patients, the analysis took 3.8 seconds.
That's incredible.
And the results confirmed these key prognostic indicators. So poor responders had about 3.9-fold higher tumor cell content, but favorable outcomes correlated really strongly with immune infiltration.
More immune cells, better outcome.
Exactly. Macrophages were 11-fold higher, and B and plasma cells were 3.7-fold higher in the patients who responded well.
And I imagine the spatial maps generated instantly would just light up, showing that tumor immune geography.
They showed this striking inverse relationship. Regions that were just dominated by tumor cells in poor responders were the exact same areas where immune cells were totally depleted. This kind of rapid throughput, it transforms how you could use spatial data for, say, real-time clinical screening.
Now let's talk about the absolute cutting edge, Visium HD. The fact that this tech can get down to 8-micron resolution, generating hundreds of thousands of spots, that let FlashDeconv do a systematic scale space exploration that just wasn't possible before.
Right, and this led to the discovery of what they call the resolution horizon.
The resolution horizon, what is that?
It's a critical concept. They were analyzing mouse small intestine, and FlashDeconv processed over 350,000 spots at the highest, 8-micron resolution in just 12 seconds.
Wow.
And by systematically analyzing that same data across 5 different resolutions, they found this crucial transition zone, right between 8 and 16 microns.
What defined that horizon? What happened there?
Geometric mixing.
Yeah.
At 8 microns, over 60% of the measurement spots were highly pure, meaning dominated by a single cell type. But as soon as they crossed that 16-micron line, that purity just collapsed. It dropped by 78%.
And why is that specific size, 10 to 15 microns, So important.
It aligns perfectly with the physical size of the intestinal epithelial cells. Once your measurement bin is bigger than the cell itself, you're just averaging multiple cell types in every single spot.
That's why the horizon matters so much. If you're analyzing data above that resolution, you're not measuring biology anymore. You're measuring a mathematical average. And that mixing, you said, it can actually invert reality, the problem of spurious co-localization.
This is the real danger zone. They looked at Paneth cells, which are only at the base of the intestinal crypts, and goblet cells, which are distributed along the whole axis.
So biologically, they're separate.
They are spatially segregated. They should show a weak negative correlation, which they do at 8 microns. They kind of exclude each other.
But once you start using those coarser bins and you start mixing these physically separated cells,
they get artificially bundled into the same measurement. And this created a mathematically induced positive correlation that peaked at 64 microns resolution.
the signal literally flipped signs.
It did. So if you had only analyzed that 64 micron data, you would have wrongly concluded that these two cell types are close collaborators, when in reality they are mutually exclusive. The coarse resolution lied to you.
And finally, because FlashDeconv was built from the ground up to preserve the structure of rare cells, it found something truly cryptic, a biological structure called the Tuft stem niche.
That's right. And this is something variance-based methods would have completely ignored. Tuft cells are a very rare epithelial cell type, and their marker genes have extremely low variance. They rank 21 percentile points lower by variance selection, so traditional methods were just systematically blind to them.
A FlashDeconv, because it looks for distinctiveness, it could see them.
It could. At 8 microns, it resolved thousands of these focal Tuft cell niches with up to 61% purity. And the revolutionary discovery was that these Tuft hotspots were enriched an astonishing 16.8-fold for intestinal stem cells.
Wow, the LGR5 marker population.
Exactly. It confirmed the specific, ultra-rare anatomical niche that's known to hold reserve stem cells.
And this structural truth is only visible at that highest resolution.
Absolutely. The maximum Tuft proportion drops from 61% at 8 microns to only 4% at 128 microns. It's the perfect storm. It's a structure that's both mathematically quiet in the gene space and spatially diluted at low resolution. You needed both the speed and the feature selection of FlashDeconv to make this discovery.
This is really the definitive story of how data science can literally clarify biological reality. FlashDeconv used this mathematical principle, distinctiveness over abundance, to save these rare critical signals, enabling this linear scalability to massive million-spot atlases.
And that ability to process data this quickly, it totally transforms resolution selection. It's no longer an arbitrary guess you make in the lab. It's a data-driven determination. The resolution horizon proves that the tissue geometry itself dictates the quality of your insight.
So if you analyze data above that horizon,
you risk generating conclusions based on mathematical artifacts, not biological truth.
A truly impressive achievement. So if all these older spatial transcriptomic studies, which were often done at much coarser resolutions, 55 microns, because of computational limits, if they've already published their findings, here's the provocative thought we'll leave you with.
I like this.
What other crucial cell interactions that we currently assume are positive collaborations because the low resolution mix their signals, what if they're actually critical mutual exclusions that we are only now, thanks to this new speed and precision, just starting to resolve?